So this segment talks about the operon and then several mechanisms of gene transfer. First, the operon. So when I went through translation and transcription, I talked about how there's some major differences between prokaryotic and eukaryotic transcription and translation. And one of the differences was I said that eukaryotic cells, it's one messenger RNA, one protein. While in prokaryotic cells, one messenger RNA can encode multiple genes in a row, and all of those genes can be transcribed and translated all at once. So an operon is a series of two or more genes that are transcribed simultaneously and translated simultaneously because they're both or all necessary for a certain process. So the reason why I have this here, I thought of this scenario when I was pregnant and I liked eating like cakes, and I made cakes all the time, and that was like my pregnancy food was cakes. So here's a scenario for you. Eukaryotes. If eukaryotes want to make a cake, they will get in their car, they'll drive to the grocery store, they'll get the box mix, and they'll come home. Then they'll get back in their car, they'll go to the grocery store, they'll get their frosting, and they'll come home. Then they'll get back in their car, they'll get the eggs, and then they'll come home. Now with eukaryotes, we want to be able to control every step along the way. So if we decide one day, I am not going to make this cake, I can do this, I'm not going to get super fat during my pregnancy, then they can stop at any point along the way. It's highly regulated. So they can choose to make the cake or not at any trip to the grocery store. At any point, they can turn around and go home and say, I'm not making this cake today. Now what prokaryotes do is prokaryotes are going to go to the grocery store, they buy everything they need, they buy the box mix, the frosting, and the eggs, and they come home and they make the cake. And they're going to make it no matter what, and there's no stopping them. Or it's really unlikely that you're going to be able to stop them. So there's a lot less control with prokaryotic cells. Now with an operon, if you need 3 separate proteins in order to perform a certain process in the cell, so let's say in order to eat the sugar arabinose, you need gene 1, gene 2, and gene 3. What eukaryotic cells would do is we would transcribe and translate gene 1 and stop. Then we would transcribe and translate gene 2 and stop. And transcribe and translate gene 3 and stop. Because at any point along the way, if a more favorable nutrient comes along, we didn't just waste a whole bunch of energy making all this protein for stuff that we're not really going to end up using in the end. We can tightly regulate and tightly control each one of those processes. But with bacteria, if you need 3 proteins in order to be able to use the sugar arabinose, you are going to transcribe and translate all 3 of those proteins at the same time. And then you're going to use arabinose, whether you like it or not. So there's benefits to having an operon in that it's more efficient, but there's also some major drawbacks because there's a lot less control in having an operon. Operons can either be inducible or repressible. Inducible means that they're usually turned off, but you can turn them on if necessary. Repressible means that they're usually turned on, but you can turn them off if necessary. And both of these have their applications when we talk about bacterial systems. The operon that we're going to focus on in this class is the Lac Operon, or the lactose operon. Now in the Lac Operon, you need 3 proteins in order to be able to efficiently utilize lactose. So we make them all at once. This is what the Lac Operon looks like. So if we picture this as a piece of DNA, a strand of DNA, there's a region on the DNA that is a cap region, then there's a promoter region, then there's an operator region, and then there's the Lac Z gene, the Lac Y gene, the Lac A gene. I'm going to talk about what each one of these components does. At the very end, there would be a termination region. So what we do is, if we're going to transcribe this, RNA polymerase is going to bind to the promoter, and it's going to copy this entire segment. And then it's going to stop once it gets to the termination region. So when it copies this whole segment here, all 3 genes are incorporated into one long messenger RNA. Now the LAC-Z gene codes for beta-galactosidase, or beta-gal. And beta-gal is necessary to break the bonds of lactose. So this is actually the digestive enzyme that breaks down lactose. The LAC-Y gene codes for permease. And permease makes the membrane permeable to lactose. The LAC-A gene codes for transacetylase, which we know it transfers acetyl groups. We don't exactly know why it's doing it in the Lac Operon. And if we knock out this gene, so we get rid of it, the Lac Operon still works fine, and we can still digest lactose. So it's really not necessary, but it's there, so we talk about it. So when we go to translate this, we have ribosomes that will turn this protein into a separate protein from this protein into a separate protein from this protein. So we have multiple ribosomes that can get on this messenger RNA, and when it reads it, it's going to just read the segment of the gene, and then there's a stop codon in here, and there's a stop codon in here, and a stop codon at the end of here. So it will actually produce 3 separate proteins from one big long messenger RNA. We have to be able to turn this thing on and off. And there's 2 ways that we regulate the Lac Operon. Number 1, this cap region. Cap binds to the cap site to bend DNA in the absence of glucose. So if RNA polymerase wants to bind, it tries to, but cap gets in the way. If cap is straight in alignment with the promoter, RNA polymerase can't bind. So what will happen is, in the absence of glucose, if glucose goes away, cap will actually bend out of the way. So why do we want to use the Lac Operon if there's no glucose? Well, glucose is the easiest sugar to use. So if glucose is present, then we're going to use glucose. We don't want to waste energy going through all this stuff if we have glucose. But if glucose goes away, then we have to have something as a sugar source. So in the absence of glucose, this cap protein will bind to the cap region and bend cap out of the way. When cap's out of the way, then RNA polymerase can bind to the promoter, and you can go through transcription of this. But there's another thing that has to happen. There has to be lactose. Why are you going to try to digest lactose when there's no lactose anywhere to be found? That would be crazy. So there's a repressor that sits in this location. And this repressor molecule only falls off if lactose attaches to it. And it's not actually lactose, it's allolactose, which is a derivative of lactose. But anyways, if lactose is present, lactose, or a derivative, can bind to this, and then this falls off. So it's no longer blocking the access to transcription of the genes. So the 2 factors that have to happen. Number 1, you can't have glucose. Number 2, you have to have lactose. If both of these conditions are met, then the Lac Operon will be turned on. The ability to utilize new nutrients, like through the Lac Operon, or the ability to be resistant to antibiotics, typically comes from acquisition of genes. Now, if you think about it, with people, our offspring is completely different from us, because our offspring is a combination of 2 different people coming together. So the sperm has its own set of alleles, the egg has its own set of alleles, and when they unite, they create a unique individual. With bacteria, they just make identical copies of themselves all day long. So in order to increase genetic variability, they have to have ways to switch up their genome. Now, one way is just through random mutation, and we'll talk about mutations in a little bit. But another way is by actually taking DNA from other bacterial cells. That's what we call bacterial recombination. So when we say recombination, we are changing the genetic makeup of the cell. There's three major methods of genetic recombination: conjugation, transformation, and transduction. Conjugation is kind of like bacterial mating. What happens during conjugation is you have a pilus, and if you remember from the bacterial lecture, I talked about a pilus being like a mini penis. So this pilus grows from one cell into the neighboring cell. Now the pilus is a tunnel that allows a replicated plasmid to go through from one cell to the next. Once it gets into the next cell, it will go back into a circular structure, and then the pilus will be severed off. So it's a way to hand out genetic material to neighboring cells. So why would bacteria do this? Well, safety in numbers. If I'm a bacterial cell and I know that I'm resistant to an antibiotic, what I'm going to do is I'm going to make a copy of my resistance gene and I'm going to hand it out to all my friends. It's kind of like that scenario, you don't have to outrun a bear, you just have to outrun your friends. So if there's a whole bunch of friends around me and the immune system is trying to eat me, I'm less likely to get eaten if there's a lot of other organisms around me. So the process of conjugation increases the genetic variability of the bacterial cells, but it's also beneficial in that it provides for safety in numbers. If you give them extra genes, they're going to survive better in the environment, and then you're going to have a larger colony. Transformation is the uptake of DNA from the environment, usually from a dead bacterial cell. So one bacterial cell dies, the DNA kind of fragments, and then a neighboring bacterial cell will suck up the DNA from the environment. Bacterial transformation is going to be talked about a lot because we're actually going to use bacterial transformation in the lab to make glow-in-the-dark bacteria. So at the end of this lecture, we go through what's involved in transformation in a lab, but this also happens out in nature. Now, transduction. Transduction involves bacterial phages. And a bacterial phage is a virus that infects bacteria. So yes, bacteria can get sick with viral infections. So here's what a bacterial phage looks like. The bacterial phage injects in its genetic material, and under normal circumstances, this genetic material hijacks the bacterial cell's enzymes and turns this bacterial cell into a virus-producing factory. Now, as it packages up new viral particles, one of them accidentally packages up bacterial DNA. So these guys that escape now will go infect other cells and they'll make other bacterial cells sick. But this one that accidentally took in bacterial DNA will go transfer the bacterial DNA to another cell. Well, now this cell is happy because it just got infected with bacterial DNA. So those are the three major methods of increasing variability or changing the genetic composition of bacterial cells.